my talk is photocatalytic application and cyclic voltammetric study of nickel pyrophosphate nanoparticles. These are the outline of my talk. So let us start with the introduction. Nickel pyrophosphate have monoclinic crystal structure having applications in energy storage devices as well as supercapacitor. It is also useful in efficient photocatalyst for hydrogen generation from water splitting applications. It is also useful for battery application and also useful as a catalyst for dye degradation. Now we are moving to the synthesis method. So to synthesize this nickel pyrophosphate nanoparticle, first we took 0.5 molar nickel chloride solution and stirred it at 200 RPM. After 30 minutes, we added surfactant Triton X100 in this homogeneous solution, and after that we stirred this solution for 20 minutes, and after that we added sodium pyrophosphate dropwise in this solution, and after that filtered it, and we got green precipitate of nickel pyrophosphate, and after that this precipitate calcined at 300 degrees Celsius in furnace, and after that we got final products, that is nickel pyrophosphate nanoparticles. These are the XRD plot, and these are the lattice constant which is well matched with the JCPDS card also. So from the XRD plot, the peak broadening indicates the nanostructure of the nature, and from the Searles formula we obtained the crystallite size, that is 15.48 nanometer. These are the TAM images. So from the TAM images, we conclude that the minimum particle size of the material, that is 33.19 nanometer, and the maximum particle size of the material, that is 57.27 nanometer, which confirm the nano nature of the sample with spherical morphology. Wastewater analysis. So these both images shows the effect of pollutant on aquatic system. Generally dyes exist in industrial wastewater and even in drinking water system, which are extremely harmful to the environment and humankind also due to their toxicity and resistance to the nature degradation. Furthermore, when the light penetrate in water will be hindered, and photosynthesis in aquatic plants will be disrupted in the presence of dyes. This will kill aquatic life and hence the effect of the ecosystem also. So for the solution, here we are doing experiment for the MB dye photo degradation. So for the methylene blue solution under the illumination of light that has energies higher than the photocatalyst band gap. So the electron from the valence band will be excited and go to the conduction band and create an electron hole pair. Will initiate a series reaction and produced hydroxide radicals and superoxide radicals. So the dye degradation leads to the conversion of organic carbon into the harmless gaseous carbon dioxide, and that nitrate, sulfate and chloride into the inorganic ions. Now for the dye degradation for UV irradiation, this is the methylene blue structure, and for UV irradiation the immobilized photocatalyst 200 mg was placed inside the beaker under the continuous stirring in dark condition, and after that we exposed the UV light on this beaker. The distance between UV light and beaker that is fixed, that is 15 centimeter, and after near about 30 to 40 minutes we get the solution is 100% degraded, and we get this colorless solution. These images shows the pure MB solution, and this shows the after adsorption solution. And one more thing, when the experiment was repeated in direct sunlight, here we are using UV light, but when these experiment are doing in direct sunlight, the decolorization took long time, that is near about 4 to 8 hours. These are the wastewater analysis result. So from this result, we conclude that the chloride concentration, chloride concentration is double in the degraded MB solution. The slight increase in sulfate, solution. They suggested that the dye is degraded in lower molecular weight. When the other hand, the phosphate the phosphate and nitrate concentration reduce in the degraded MB solution. This leads to conclude that the photodegradation affects chloride, sulfate, phosphate, and nitrate concentration. These are the TOC, total organic carbon study. So from the total organic carbon study, we can observe that the mineralization efficiency is continuously increases as per the irradiation time is increases. That is a very good result. Ion chromatography. So ion chromatography is carried out to identify the nitrate, sulfate, and chloride into the inorganic ion present in the sample. So from the study confirmed that the dye degraded in lower molecular weight dye, and from the study the confirmed that the nitrate and chloride ions increased on irradiation. These are the all, and these results are also correspond to the wastewater analysis data also. UV spectral study. So from the UV spectral study, the pure MB solution exhibit sharp absorption peak at 246, 292, 613, and 667 nanometer, while the photodegradation MB solution sample exhibits no absorption peak. So the extent of degradation of MB under UV light irradiation was found, that is 95.06%. These are the electrochemical impedance study. So this is the Nyquist plot at different frequency range, and the value of grain resistance and grain capacitance at the particular frequency range is mentioned in this table. Cyclic voltammetry. So to determine electrochemical capacitive properties of nickel pyrophosphate nanomaterial, cyclic voltammetry experiment within a 0 to 0.6 volt range at a skein rate from 5 to 100 millivolt per second were performed in 3 molar KOH electrolyte at room temperature. On the basis of their energy storage mechanism, supercapacitors can be classified as electrochemical double layer capacitor and pseudo capacitors. So for that, this figure shows the EDLC double layer capacitor. So in EDLC double layer capacitor of ions is created at the interface of the electrode surface and the electrolyte, while in pseudo capacitor energy is stored through fast super redox reaction, which take place at the electrode electrolyte interface. These are the type A, type B, and type C capacitor. So this shows the current versus potential plot, and from this plot we conclude that the which type of behavior of our capacitor. Okay. So cyclic voltammetry. This is the current versus voltage plot. Nanostructure electrodes is based on the surface redox mechanism of nickel 2 plus to nickel 3 plus at the surface, and the shapes are different from that of EDLC. The shape of these loop are different from the EDLC, suggesting that the capacity mainly results from the pseudo capacitive capacitance. With increasing the skein rate, the peak current shows a linear increment, denoting high electron swift and ions in the nanoparticles. Capacitance versus specific capacitance versus skein rate plot. So from this plot, maximum specific capacitance found to be 406.33 at the skein rate of 5 millivolt per second, while the 100 while the skein rate is 100, at that time the value of specific capacitance is 49.5. The specific capacitance decrease with increase the skein rate, may be due to the rapid mobility of the charged particles. Galvanostatic charge discharge. So from this graph, we calculate the value of specific energy and specific power that are already mentioned. In this table. So the specific energy and specific power are two key factors for evaluating the power applications of electrochemical supercapacitors. A good electrochemical supercapacitor can provide a high energy density and specific capacitance. Now finally, I conclude my talk. Nickel pyrophosphate nanoparticle calcined at 300 degrees Celsius. Photocatalytic activities were evaluated by decolorization and degradation of 10 ppm of MB solution, which was prepared by adding 10 mg methylene blue in 1000 ml water. From wastewater analysis, we conclude that the concentration of chloride is almost double compared to the degraded MB solution and pure MB solution. Thus, the drastic change occurring in chloride concentration suggests that the dye was degraded in lower molecular weight. From TOC study, uh, at the beginning of reaction, the TOC value slowly decreased and then decreased rapidly. The mineralization efficiency increased as the irradiation time is increased after 90 minutes of the constant stirring of MB dye and the catalyst solution under UV light mineralization. The efficiency was found to be near about 83.1%. Ion chromatography technique suggested that the dye degraded in lower molecular weight. And from CV analysis, we conclude that this nickel pyrophosphate exhibit both Faradic and pseudocapacitor behavior and and can be used as a supercapacitor. Finally, I acknowledge acknowledgements and thank you for your kind
This
attention.
paper, just now I told that it explores the topic of impact of nanosilica additions on behavior of the cementitious systems and cement-based materials, unveiling its potential to the properties enhancement and improve durability, simply all these properties, and also insights on the systems and implications, how it can be implied for the construction and infrastructure development. Now, definition of nanosilica additions refer to the introduction of nanoscale silica particle. What nanoscale means 10 to the power -9 meter, as per the American Concrete Institute definition of nanoscale. These particles can significantly impact the behavior of cement materials by enhancing the mechanical properties I just told. With a small size and unique properties, nanosilica additions hold immense potential for revolutionizing this construction and infrastructure development. The importance of this system, cementitious systems form the backbone of construction and development. Understanding the impact of nanosilica addition on the cementitious system is crucial to harness their full potential. These additions can enhance the mechanical properties, reduce shrinkage, and improve the durability of the cement material. By incorporating nanosilica, we can create more resilient and sustainable structures that can withstand the challenge of the time. Now, another behavior of cement system. The addition of nanosilica addition to cement system is strategic behavior that can greatly impact the performance of structures through the influence of hydration kinetics, strength development, post-structure development, and durability properties. By studying the behavior of cement system with nanosilica, we will optimize the design and construction of infrastructure and leading to more sustainable and resilient structures. The overview of nanosilica additions offer numerous advantages. They enhance the early stage development for the fast track projects, improve the workability of the system, cement system, reduce the permeability of cement system, increase the overall durability of the cement structures. Under the potential of nanosilica addition allows us to create more resilient and sustainable structure with long-term performance and minimize the maintenance cost. The cost factor is also important. I will come in. Excellent studies have been conducted in our lab.
Slide is not moving.
Moving?
Yeah, you have to click the left
I
side.
have to click, no?
Yeah.
Now.
Now it is 6. Experiment.
Got it. Yeah. The studies conducted focus on analyzing the effects of adding the nanosilica content from 0% to 1.5% with respect to the weight of cement, mixing methods through internal curing and also external curing, condition that normal temperature curing in the water lab, and the other factors on the mechanical properties, microstructure, and long-term performance of the cement composites. The result of the studies contribute to understanding of nanosilica's potential as a valuable addition in cement-based materials. Now, coming to result, key finding and results, what we have got. Now it is coming? Yeah. Improve the compressive strength of cementitious system, increase the durability of cementitious system, improve the microstructure of cementitious system, enhance the bond strength in the cementitious system. Proper procedure helps in optimization of the nanosilica addition, because optimizing is very important, because you cannot just you add the nanosilica at any percentage, because there is an optimum percent. You should find out that the long-term performance to demonstrate the sustainable benefits of nanosilica addition. And the findings highlight the potential of nanosilica as a valuable additive in enhancing the behavior and performance of cementitious systems. Now the reaction envelope. In an ordinary cementitious system, what happens? C3A, there are in cement there are four compounds: C3S, C2S, C3A, and C4A. Tricalcium silicate, tricalcium aluminate, and tetracalcium aluminate ferrite. Among these, C3S is the main more than 50% of the cement is C3S, and it it reacts with water to form calcium hydroxide. See this the second reaction. This calcium and this CSH, calcium silicate hydrate, which is a crystalline compound and which is responsible for the strength of the cementitious system. This calcium hydroxide reacts with the nanosilica if present, if added, and gives us additional calcium silicate hydrate, the crystalline compound which is responsible for the strength in cementitious system. So these two nucleation reaction, then the nucleation reaction comes, and it also adds up this additional calcium silicate hydrate. From this ion is formed, and from this ion is formed from the nanosilica, and this one goes on. So we get additional calcium silicate hydrate, the compound responsible for the strength development. Now what I got my nanosilica from BChems. BChem active nano, it's a colloidal type of nanosilica, 35 to 40% solid nanosilica. The particle size diameter varied from 50 to 40 nanometer, and the specific gravity 1.08 to 1.31. This is XRD, which shows that it is not crystalline, it is amorphous, no peak was found. And the test performed: 3 days, test compressive strength, 7 days, 28 days, and 365 long term. What we found, we added 0.5, 0.75, 1.25, and 1.5% nanosilica with respect to the cement. And what cement we get as per IS code 4031, that cement part I will coming. And what we found, 26% gain, 27% gain at 3 days. So 7 days, 31.54% gain with respect to the normal addition of nanosilica. And 28 days, 32.55% gain, the 0.75% nanosilica was by optimized. Sorry. So then, is it coming? Yeah. This is the graph, at various days, 7, 20, 65 days, and the various proportion of nanosilica added. See, the 0.75% is going highest, giving highest strength. Microstructure analysis coming. This is the cement without nanosilica. See, the cracks is there, and also uneven, some parts is white, some parts is without white. White means the crystalline formation. And the next thing is with nanosilica, see some uniformity of white particles, that is CSH, calcium silicate hydrate, crystalline formation is there, with lesser amount of cracks, and also photoline is there. So we can say the much defined form. Now the cost economics comparison, cement part 1% by weight of cement, sand 3% by weight of cement, polycarboxylate ether, we added an admixture, that is 1% by weight of cement, and taking colloidal nanosilica, we get this is the silica film composite, and these rates I got from this paper, cement concrete composite 2012 construction building materials journal, and construction building another journal of 2013. These two helped me. These three journal papers helped me to get the rates. And I find that 10 to 11% decrease in total cost in my colloidal nanosilica composite compared to the silica film composite, because the percentage of this 34.26 silica film kg we are added, and that paper had added, and I added 4.28 kg of nanosilica to produce 1 meter cube of composite. So 4.28 and 34.2, so much less, and that's why the cost economics came into picture. Much could be done in much lesser cost compared to the silica film composite. Benefits of application of nanosilica addition offer numerous benefits of application. Just showed improved compressive strength, enhanced durability, reduced permeability, increased resistance to chloride papers, improved microstructure, bond strength, and also structure indicate. Proper mixing methods and curing conditions are essential for optimal results. I was just telling 0.75% nanosilica was my optimized dosage. This could not be attained in another lab, or if you play with the diameter of the nanosilica, the optimal dosage may vary. So, I mean, the proper mixing methods and the external conditions should be proper to get the actual optimized result. Huh? A long-term performance change demonstrates sustained benefits. Sustained means why? Because I am getting much less cost and much greater strength. I am getting much valuable benefits, property benefits with lesser cost. That's why it is most sustainable. So, nanosilica is valuable additive in enhancing the behavior and performance of cemented systems. Now we have challenges. What challenges, with dispersion and agglomeration of nanosilica? I was just saying, because 1.5% nanosilica, much greater percentage compared to the 0.75 what optimal I got, I am not getting so good result. So what is the reason? That's why this dispersion and agglomeration of nanosilica particles came into picture, and that's the challenge in implementing nanosilica in the large scale projects for for high performance concrete and all those cementing systems. So this should be clearly evaluated to get the sustainable benefits of this nanosilica added in cemented systems. So conclusion, have great potential in improving the behavior and performance of cemented systems. However, it is important to address the challenges and limitations associated with these activities. Careful consideration should be given to issues such as particle dispersion, compatibility with other activities, what are admission of the polycarbonate ether, cost and long-term sustainability. By weighing these factors, engineers and researchers can make informed decisions about incorporating nanosilica in large-scale projects. So this is nanosilica, microstructure modification, durability properties, all these are improved. And their circle, if one is improved, other property gets improved automatically, by improving the total microstructure cemented systems. So, thank you. Any questions we have, Vikas?
Good afternoon, everyone. I am Dr. Manojji Deka, Department of Chemistry, BP Solia College, Assam. This college is affiliated to Guwahati University. And firstly, I would like to thank the organizing committee to give me this opportunity to present here. So my topic is investigating optical properties of biomass waste derived carbon quantum dots prepared by hydrothermal method. So in our laboratory, we are mostly working on some waste material, and this waste material we converted to quantum dots nanomaterial as well as activated carbon. So before going to this in detail, so introduction. So these are the electrons of carbon, graphene quantum dots, carbon dots, CNT, fullerene, diamond, etc. So their hybridization are different and their dimension are also different. So our research interest mainly focused on these three nanomaterial: graphene, two-dimensional, 2D sp2 hybridized, then these graphene quantum dots and these carbon dots. So what is carbon dots? So carbon dots can be generally defined as a quasi zero-D carbon-based material with size below 10 nanometer, and it shows beautiful fluorescence property, and this is their intrinsic property. And this was first reported in 2004 when carbon nanomaterial with fluorescent property were first reported, accidentally obtained from the purification of singular CNT. So these carbon dots can be classified into three category. One is graphene quantum dots, one is carbon quantum dots, and other is carbonized polymer dots. So this classification is based on their mechanism of formation, nanostructure, and their properties. So this is the classification of carbon dots. So graphene quantum dots all are zero-dimensional. So graphene quantum dots are mainly from graphitic origin, like fullerene, graphite, carbon nanotube, graphene oxide, and these are mainly synthesized by cutting or ultrasonation, these methods. Then these carbon quantum dots can be synthesized from any biomass, plant and animal origin, and the process are carbonization, thermal and hydrothermal. And these carbonized polymer dots are synthesized from polymers having carbon backbone. So all these things are grouped in carbon dots. So for graphene quantum dots, quantum size graphene fragments, and they are zero-dimensional, and diameter is usually less than 100 nanometer. They are arranged in a planar and hexagonal lattice, and it is mainly composed of sp2 bonded equivalent carbon atoms with single sigma bond. Then For carbon quantum dots, not necessarily quantum size graphene fragments, and it is composed of sp3 bonded equivalent carbon atoms, and they are also zero dimensional, or arranged may not be in a planar and hexagonal lattice, and diameter is usually less than 10 nanometer. So this is the difference between these graphene quantum dots and this carbon quantum dots. So the quantum dots may be synthesized by various methods like hydrothermal, laser ablation, chemical vapor deposition, ultrasonic shearing, then chemical exfoliation, etc. So we mainly use these methods: chemical exfoliation, hydrothermal, and for graphene, we have used this chemical vapor deposition method. So these are some properties of an application of carbon dots, like they are high quantum in biocompatibility, eco-friendliness, and they have strong absorption, good fluorescence. Then this can be used in biomedicine, sensor, catalysis, and many more, energy storage application, optoelectronics, etc. So these biomass derived carbon dots can be plant or animal origin. So for plant origin, maybe from fruits, vegetables, leaves, flower parts, etc. These are already reported. And for animal derived from eggs, cow milk, body waste like urine, human hair, gram cell, tap cell, etc. So, and many more reports are there for synthesis of these carbon dots from biomass having plant and animal origin. So our laboratory mostly working on these three waste material. These are very readily available in Assam, and they are water hyacinth waste, then tea waste, and this Areca nut husks. And this we have synthesized carbon dots and activated carbon from this waste material. So in this presentation, I would like to give one work of water hyacinth derived carbon quantum dots for sensing some herbicide. So carbon dots, so this method we have, in this work we have synthesized this carbon quantum dots from this water hyacinth, and this is the procedure. So water hyacinth, this is the collected water hyacinth from our college campus, then dried in sunlight in 3 days. These are then remove the chlorophyll, then it is dried, and it is powder of this water hyacinth. Then we sieving this powder, and this impregnated in phosphoric acid for 24 hours for activation, and then it is heated to 150 degrees Celsius. After that, it is dispersed in water and sonicated and centrifugation, and our desired product is ready for the characterization. So here, most importantly, we have used this phosphoric acid as activating agent, and it is the first report from water hyacinth to synthesize this carbon dots. So this is the characterization. So from same images, it is seen that the particle size is below 10 nanometer, and dynamic light scattering measurement also shows this result that size are below 10 nanometer, and X-ray spectra shows that amorphous behavior, and the sharp peaks are disappeared after carbonization or hydrothermal treatment. Then this is the TEM images, and 10 images shows the particle size distribution below 10 nanometer and in approximately 5 to 6 nanometer. So after this characterization, we are confirmed that the synthesis of these carbon dots are successful. Then we have also studied their very interesting property, photoluminescence property of this synthesized quantum dots. Then from UV, it is seen that the one peak is there in 280 nanometer, and this may be due to some pi to pi star transition of this carbon dots, and these carbon dots are highly oxygenated. So, and the PL behavior shows that it shows excitation dependent PL behavior. So increase in excitation wavelength, the peaks are shifted, and the maximum emission is observed at 300 nanometer excitation wavelength, and it is found to be 370 nanometer emission at 370 nanometer. And this is the dispersion in UV light. It is shows green fluorescent, and this is images for normal light. So this photoluminescence behavior or excitation dependent behavior may be due to some reason like size effect or functionality present in the carbon dot surface, or maybe some quantum confinement effect. So this is the most important property of carbon quantum dots, having this excitation dependent photoluminescence or fluorescence behavior. So this is the structural information. Then from this FTR spectra, we are satisfied that the surface of the carbon dots are oxygenated, and the peaks intensity are decreased, maybe due to the hydrothermal protocol for the preparation of carbon dots. The peak intensity are decreased, and some oxygenated functional groups may be added, appeared after the carbonization process. Then we have studied this fluorescence property to sense or to detect some herbicide, and we choose a herbicide petrylachlor. So for that purpose, when we added this herbicide to this fluorescence quantum dot, we seen interestingly turn on fluorescence, and this is the PL curve, and this is the Stern-Volmar plot. From this curve, the linear regression constant is found to be 0.9847, and it shows good linear response, and the shows closeness to the fitted regression value. And then we have calculated the minimum detection limit, and it is found to be 2.9 micromolar and equivalent to 0.9 mg per liter. And from literature, it is seen that the toxicity limit of the petrylachlor is the range of 1 to 8 mg per liter. So hence the carbon dot based PL sensor can be easily detect this herbicide if the toxicity limit lies between this range. So after that, we have the selectivity study of our sensor, and it shows good.
Dr. Manas, please conclude your work.
Okay. Petrylachlor, it shows very high selectivity, and we put forward a single mechanism to this. Maybe due to the electron transfer mechanism is responsible for this turn on fluorescence for this carbon quantum dot synthesis from the water hyacinth waste. So this is, we are also working on some carbon dot synthesis from carbon dots from T waste and activated carbon. This work is going on, and these are some characterizations which shows the blue fluorescence, and this also shows some excitation dependent PL property of the synthesis carbon dots from T waste. So in summary, we have successfully synthesized the carbon dots from this water hyacinth and T, and from water hyacinth derived carbon dot shows green fluorescence and size below 10 nanometer, and it is oxygenated in nature. And both series possess excitation dependent PL behavior like other carbon nanometers already reported in literature. So, and we have detected this herbicide and petrylachlor, and it is detection limit is found to be 2.9 micromolar. So this shows highly selective, and we also use for real soil sample contaminated with this herbicide, and we got good result. So I would like to thank our principal and our HOD for giving me this opportunity to complete this work. And I would also like to Dr. Devashish Chaudhary sir, my supervisor and group leader of Material Nanochemistry Laboratory, ISST Guwahati, for the help and support. These are my publications. Thank you.
Thank you, Dr. Manas. So anybody has query? Any questions, query from the Dr. Manas? From the audience side, please.
My paper entitled Encapsulation of Bioactive Organosulfur Compounds in the Solid Lipid Nanoparticles to Combat the Multi-Drug Resistance Pathogen. So as we know, the nanotechnology is increasingly applied in the agriculture and food science, particularly in the nano-encapsulating bioactive compounds, to enhance their stability, solubility, and viability while protecting them from the degradation. I'm using the allyl sulfide and adyl sulfide extracted from garlic. Those are the organosulfur compounds of the garlic, those already having the antimicrobial properties. But the problem with these organosulfur compounds is that they are unstable in nature and their viability in the nature is very less. So I'm trying to encapsulate these nanoparticles. I'm trying to encapsulate these organosulfur compounds in the solid lipid nanoparticles to stabilize them and increase the viability in the nature. And These organosulfur compounds loaded nanoparticles that is were prepared by the emulsification solvent diffusion techniques coupled with a low temperature solidification with a slight modification by Porves et al. Now, I've characterized these allyl sulfide solid lipid nanoparticles and the diallyl disulfide solid lipid nanoparticles using the UV visual spectroscopy, FTIR, zeta sizing PDI, and TEM. Now, the UV spectrophotometric analysis shows here that the UV spectra for the DS, SLN, SLN, and DS were recorded. In the UV spectrum, the DS, a distinct peak at the 260 nanometers were identified, while the two separate peaks at 230 and 270 nanometers were observed in the SLN. And the UV spectrum of the DS, SLN exhibit a broader peak suggesting the successful encapsulation of DS in the SLN. This is the FTIR data of the allyl sulfide SLN and the diallyl disulfide SLN. So the FTIR spectra of the lipid nanoparticles and the drug loaded with the lipid nanoparticles reveals distinct differences in the peak at 1495.40, 1348, 1295, and 1097 centimeter inverse in the allyl sulfide solid lipid nanoparticles. And there's a distinct differences in the broader peaks at 1222, 23, 1151, 1106, and 1040 in the DADS SLN, suggesting that these drugs are successfully encapsulated in the nanoparticles as we taken the solid lipid nanoparticles as a standard. So these change in the peaks possibly indicating the change in the lipid and the drug interactions. So this is the size distribution of the TEM and the TEM analysis. So the TEM result shows here that the allyl sulfide SLN and the diallyl disulfide SLN were spherical in shape, and the size of both the synthesized solid lipid nanoparticles under the TEM correlate with the zeta size. So the average zeta size of the diallyl disulfide solid lipid nanoparticles was found to be 211.2 nanometers, and the PDI of the SLN was found to be 0.198, whereas for the similar for the allyl sulfide solid lipid nanoparticles, the zeta size was found to be 275 nanometers, and the PDI of the SLN was found to be 0.187. So this is how we calculate the drug loading and encapsulation efficiency. So this is the formula we have used, and these are the DADS standard curves and allyl sulfide standard curves present here. So the encapsulation efficiency and the drug loading efficiency was determined to be as high as 83.30 and 17.47 for the allyl sulfide solid lipid nanoparticle respectively, and 79.15 and 15.78 for the DADS SLN respectively as the encapsulation and the drug loading efficiency. Now, we have tried to find out the antibacterial activities of synthesized nanoparticles. So these are some of the antibacterial assay against the Bacillus cereus multi-drug resistance bacteria. So as the allyl sulfide solid lipid nanoparticles and DADS, that is diallyl disulfide nanoparticles, were tested against the Bacillus cereus, shows the inhibitory effect against these multi-drug resistance pathogens. So here, here's the evaluation of minimum inhibitory concentration of these synthesized nanoparticles was found to be 0.0780% for the diallyl disulfide solid lipid nanoparticles, and it is to be 0.156% for the allyl sulfide solid lipid nanoparticles. Now, another antibacterial assay, that is Kirby-Boyer method, is used for the determining the zone of inhibition of these synthesized solid lipid nanoparticles. And there's the curve. We have taken the 15 mg per well of the drug in the first figure, and the second is 1 mg per well, 2 mg per well, and this is 3 mg per well. And the control is and it is compared with the control that is taken the conventional antibiotic, that is rifampicin. It's 10 mg per well. This is the growth inhibition curve of the Bacillus cereus. When you perform the growth curve of the Bacillus cereus with the both control and the organosulfur compound encapsulated solid lipid nanoparticle treatments, so there's a significant inhibition was observed in the solid lipid nanoparticle treatment compared to the control. Here the strong antibacterial effect of the organosulfur compound encapsulated solid lipid nanoparticles due to the controlled release mechanism. So in this figure, the green dot shows that the control, and the purple shows the blank, whereas the diallyl sulfide and the allyl sulfide solid lipid nanoparticle are comparatively are very less, shows very less growth. Those bacteria treated with the allyl sulfide and diallyl disulfide solid lipid nanoparticle shows comparatively less growth to the comparative the control, and slightly higher than the blink. So these are the SAM analysis and bacterial viability assay. These are used for checking the mode of action of the synthesized nanoparticles, as the SAM analysis was conducted to evaluate the morphological alternation and the cell wall disruptions and the Bacillus cereus bacteria when it is treated with the nanoparticles. So the first figure A shows the control, that is untreated bacteria. Second is the allyl sulfide solid lipid nanoparticle treatment that shows the disrupted bacteria in the figure. The red arrow shows the disrupted bacteria and the yellow shows the normal bacteria. Whereas the diallyl disulfide and the third figure shows the diallyl disulfide treated solid lipid nanoparticles. The next figure is here the bacterial viability assay. We are using here propydium iodide fluorescence to employ the bacterial cell membrane permeabilization and DNA binding without organosulfur compound solid lipid nanoparticle treatments. So here in the control, the figure shows the less amount of RFP bacteria, that is because the propydium iodide is a intercalating dye that binds to the basically the nuclear material. So it is impermeable to the cell membrane, but when the bacteria are dyed, it goes inside the bacteria and it binds to the basically nuclear material of the bacteria. So here in the control, there's a less amount of bacteria are shown due to they're living in nature, and so the less amount of bacteria are shown as comparative to the control. In the allyl sulfide treated solid lipid nanoparticles, the bacteria are increased in number. Then the number of bacteria that die are increased in number. Yes.
Conclude, please conclude.
Okay. Due to increase in number due to the it is effective in nature. Here's the conclusion of my study, that the encapsulation efficiency and drug loading efficiencies are used up to this 83.30 and 17.47 for the ASSLN, and it is 79.15 and 15.78 for the DADSSLN. So the synthesized organosulfur compound encapsulated solid lipid nanoparticles were found effective against the MDR strains of the Bacillus cereus and various other microorganisms like Pseudomonas and E. coli, etc. These nanoparticles exhibit the significant antimicrobial activity and could be used to treat the various pathological condition, addressing the growing concern of antibiotic resistance and advancing the field of medical biotechnology. This is the acknowledgement and references of my research.
Thank you, sir. My topic is nanomaterials-based electrochemical sensor for paraphenylene diamine. As we know that paraphenylene diamine is basically azo dye, which is extensively used in the hair dyes, and millions of people are using for their hair coloring and tattoos, etc. So basically paraphenylene diamine is also utilized in polymer industries, textile industries, and pigments productions also. But if a long time we are in touch this constituents of the product, so basically it causes a number of ailments in human beings like severe dermatitis, eye irritation, tremors, coma, mutagenicity, and sometimes it also has been found that it has some cancerous symptoms. So if basically if manufacturing industries are discharging their waters and all that contaminate the local water bodies and causes hazardous effects to humans as well as in aquatic animals. So therefore the detection and quantification of this paraphenylene diamine is an interesting and challenging task to monitor the concentrations in hair dye and water samples. So number of methods are there for the detection method, but basically we are working on the developing of electrochemical sensors using the different nanocomposite materials. So we prefer the electrochemical sensing method because of its cost effective, easy to use, fast, and miniaturized. It can be miniaturized, and it is having high sensitivity and selectivity. So we develop basically different types of nanocomposites, and we modify the electrodes, and those electrodes we use in the development of electrochemical sensors. So basically whatever the nanocomposite materials we develop, that should have the property to sense in the form of either oxidation or reduction process to different types of analytes. So basically here also we use the different types of the nanocomposite material. So for the detection of paraphenylene diamine, very few works have been there in the literature. these only only few works have been I found. So that's why I was interested to do further the work for the development of the electrochemical sensor, which can sense a very small amount of the paraphenylene diamine if it is present in any hair dye sample or in water samples. So we have utilized the graphene oxide because we know that the graphene is having extraordinary properties like large surface to volume ratio, surface adsorption, excellent electrical conductivity, and high electrical mobility. So basically, that's why the graphene is utilized in the sensing application. So I have also utilized this material. So for the preparation of the nanocomposite materials, I have functionalized the graphene oxide with the ionic liquid first, and then we have combined with the electrochemically prepared copper Ag bimetallic nanocomposite, and this is the total. You can see the preparation methods for the nanocomposite material, and this nanocomposite material has been formed in the form of film on the electrode, and then electrode has been utilized for the electrochemical sensor, and we have used the electrochemical workstation for these electrochemical studies. So first we have characterized the material with the FTIR and Raman and SEM and other techniques. So in the FTIR spectra, we have a little bit confirmed that okay, yes, ionic liquid have been functionalized on the GO, and then CuAg combination form the composite, which has been confirmed. Then Raman spectra, when we have studied, we have found that for the GO, if we calculate the ID by Ig ratio, so that is 0.98. For the ionic liquid, so it is 1, and for the final composite, it is 1.0. So we know that if the ID by Ig ratio increases, it means the defect is there, and it means there is that confirm the formation of the nanocomposite by the combination of the different components. So in the SEM characterization, we also found here just okay, ionic liquid functionalized GO shows like a layered structure, but when we introduce the CuAg nanoparticles in this, so the more exfoliation comes by, because we know that GO is a little bit few layers is there in the GO, but when it is functionalized with ionic liquid and further with the CuAg nanoparticle, then the distance between the layers increases, and in this way it becomes more exfoliated, and in this way it becomes more electrical conducting. And EDEX of the composite material also shows the different elements present in the nanocomposite. Further, we have characterized with the TEM technique, and here also we in the ionic liquid functionalized GO, we found that there is a crinkled silk-like sheets is there, but after the mixing of the CuAg nanoparticle, there is a granular porous surfaces are found, and you see here, you can see that ionic liquid GO is just wrapped over the CuAg nanoparticles. So it means the confirmed formation of the nanocomposite occurs here. In the diffraction pattern also shows that polycrystalline phase of the material, and it also shows the different phases of the CuAg nanoparticles, bimetallic nanoparticles. And when we see the nanoparticle size distribution, so average particle size distribution, we found that 33 nanometer plus minus 2.4 nanometer. After the characterization of this nanocomposite material, so we have formed a thin film of this nanocomposite material by dispersing in a aqueous medium, and then with that electrode have been utilized for further electrochemical studies. So first we have utilized this technique, the differential pulse voltammetric technique, and here first we have taken the different studies without addition of the paraphenylene diamine, after addition of the paraphenylene diamine, right? So in this way we have found that okay, in the case of the nanocomposite, final nanocomposite modified electrode, the current is very high. It means it is highest electroconductive as compared to the other modified electrodes. So we have taken this modified electrode for further studies, and also we have checked whether the other isomers of the paraphenylene diamine also can interfere or not. So we have found that no, there is no any interference of other isomers of the paraphenylene diamine have been occurred here, because all the isomers gives the different peaks at the different potentials. So it means paraphenyl diamine only gives here. So it means there is a selectivity, and there is no any selectivity issue is there with the electrode. So further, we have done the cyclic voltammogram of the different at the different scan rates, and then finally we have taken the current differential pulse voltammogram with the different concentrations from 0.018 micromolar to 22 micromolar, and we have found that okay, yes, this in the range of this concentration 0.018 to 22 micromolar, we found a linear calibration plot, and through which we easily can calculate the limit of detection, the lowest limit of detection, which can be determined through our modified electrode, that is 3.9 nanomolar. So we have also calculated the effective surface area for different electrodes. So if we modify the only with the graphene oxide, so surface area was 1 into 10 to the power minus 7 mole per centimeter square. But if we modify it with the ionic liquid functionalized GO on the glassy carbon electrode, then in the surface area was increased little bit, 1.4 into 10 minus 7. But if we take the combination of the ionic liquid functionalized GO and COAG bimetallic nanoparticle, and we modify on the glassy carbon electrode, then active surface area we found 2.9 into 10 to the power minus 7. It means by the modification of this final composite nanocomposite, the active surface area was enhanced, and it helps in electrocatalyzing the paraphenylene diamine at lower potential as compared to orthoparaphenylene diamine and metaphenylene diamine, which results there is the less chances of the other interferences of other isomers, and through which we can efficiently detect the paraphenylene diamine. We have also optimized the different optimization conditions by in choosing of the different ratios of the material. So if we take the amount of GO, so we have optimized the different amount of the graphene oxide for the preparation of the nanocomposite material. So if we take the 0.5 milligram per ml, so it is suitable. Why? Because if we take more than this, if we have taken this more than this 0.5 milligram per ml, so what happened actually, the current was not you can say there is a resistance created, right? So that's why we have optimized the amounts of the GO also here. So it was that 0.5 milligram per ml is okay for the preparation of the nanocomposite here. And in the case of amount of ionic liquid, how much amount of ionic liquid we have to prefer, so we have optimized different amounts of the ionic liquid while the preparation of nanocomposite. So it is found that if 5 ml of ionic liquid is preferred, so the current is maximum, and after that if the volume of the ionic liquid we increase, so the current is a little bit steady, so it means 5 ml is okay. And the third, when we coat the film of the prepared nanocomposite dispersion on the electrode, then the amount of how much amount of the nanocomposite dispersed nanocomposite should be taken. So we have also optimized this. So 10 microliter is preferred for making the film on the glassy carbon electrode, and in this condition, the maximum current we found. So we have also taken key ratio, key how, while the preparation of nanocomposite, key what the ratio should be there of the ionic liquid functional GO and CuAg bimetallic nanoparticle. So in this here, we have taken the different ratios and optimized that key 1:4 ratio gives the highest maximum current. So while preparation of nanocomposite, we have taken the 1:4 ratio of the ILGO and CuAg bimetallic nanoparticle. In the study of the detection of the paraphenyl diamine, we have also optimized the different pH. In the acidic condition, what happens, in the basic condition, what happens, or in the neutral condition, what happens. So we found that okay, at the neutral conditions, it gives the maximum current. So in this condition, we can easily detect the different concentrations responses. So that's why we have taken the 0.1 molar phosphate buffer saline here, pH 7, as an electrolyte for the detection of the paraphenyl diamine in samples. And we also have checked firstly, we have already checked the different isomers of the interference of the different isomers while the preparation of the paraphenyl diamine, but we also detect some other constituents which may be present in hair dyes like resorcinol, hydroquinone, hydrogen peroxide, ammonia. So they are also used in the hair dyes. So we have also checked whether any effect of or any interference of these components which may be present in the dye is there or not. So we have found that yes, there is negligible interference of other constituents which may be present in the dyes. So for the real-time experiment, we have done some recovery experiment, and we have taken these 3 samples: distilled water, drinking water, and hair dye sample, which we have collected from salon, from the market, and from the barber shop. And then we have studied this recovery experiment. And in here, the data is shown here. In the distilled water, the recovery was 98.4%. For the different concentration, for 5, if added to 98.4, if 10 is 98.3. So in the drinking water, it is from 97.8 to 98.6. And the real hair dye samples which we have collected from the barber shop, so the recovery experiment is recovery result is 99.6 to 100.3. So it means we have compared the results also with the HPLC data also. So we have found that yes, our modified electrode is just comparable, comparative to the HPLC method. So, but HPLC is a very costly method. So if we prepare a little bit electrode type device, so easily we can get immediately how much concentration of a paraphenyl diamine is any kind of sample. We don't need to go for a large time sample preparation and other things. So we have checked the reproducibility test with our modified electrode also.
Try to wind up within 1 or 2.
Yes, sir. For 10 days, same electrode we have utilized for the detection, and we have found the excellent result with RSD 1.4%. So these are the reported results in which we have found that our modified electrode gives a best result, because the detection of limit was 3.9 nanomolar in the range of 0.018 to 22 micromolar. This work has been already published in the Journal of Electroanalytical Chemistry. So I acknowledge all the organizers, and these are the references which we have utilized for this work.